Hello everyone. In this context, today we'll learn chapter 20, Locomotion and Movement. As you all know, movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement. Movements of cilia, flagella, and tentacles are shown by many organisms. Human beings can move limbs, jaws, eyelids, tongue, etc. Some of the movements result in a change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, swimming, all are some forms of locomotory movements. Locomotory structures need not be different from those affecting other types of movements. For example, in paramecium, cilia helps in the movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for changes in body postures and locomotion as well. So all these above observations suggest that movements and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movements, but all movements are not locomotions. Methods of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic conditions, or to escape from enemies or predators. Cells of the human body exhibit three main types of movements, namely amoeboid movement, ciliary movement, and muscular movement. Some specialized cells in our body, like macrophages and leukocytes in blood, exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of protoplasm, for example in amoeba. Cytoskeletal elements like microfilaments are also involved in amoeboid movement. Ciliary movement occurs in most of our internal tubular organs, which are lined by ciliated epithelium. The coordinated movements of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. Movement of our limbs, jaws, tongue, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human beings and majority of multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural systems. So, in this chapter, you learn about the type of muscles, the structure, mechanism of the contraction, and important aspects of the skeletal system. Also, there are flagellar movement shown by sperm and euglena. Flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protozoans like euglena. Now let's learn about human muscular system. You have studied in chapter 8 that the cilia and flagella are the outgrowths of the cell membrane. As I told, flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protozoans like euglena. So muscle, it is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscles. They have special properties like excitability, contractility, extensibility, and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscles are identified: skeletal, visceral, and cardiac. Skeletal muscles are attached to a skeleton or are closely associated with the skeletal components of the body. They have a striped appearance under the microscope and hence are called striated muscles. As their activities are under the voluntary control of the nervous system, they are known as voluntary muscles too. They are primarily involved in locomotory actions and changes of body postures. Visceral muscles are located in the inner walls of hollow visceral organs of the body, like the alimentary canal, reproductive tract, etc. They do not exhibit any striations and are smooth in appearance. Hence, they are called smooth muscles or non-striated muscles. Their activities are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. Now, as the name suggests, cardiac muscles are the muscles of heart. Many cardiac muscle cells assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature, as the nervous system does not control their activities directly. In this table, you can see comparison of muscles at a glance. In each column, 3 different types of muscles characteristics are given, or you can see the comparison of skeletal muscle, visceral muscles and cardiac muscles in this table. Now let's see the structure of striated muscles. Okay, skeletal muscle is made up of many bundles, which are known as fascicles. Fascicles are held together by collagenous connective tissue layer known as fascia. Each fascicle contains many muscle fibers. Muscle fibers are lined by plasma membrane known as sarcolemma. So, plasma membrane of muscle fibers are known as sarcolemma. So, muscle fibers, they are lined by sarcolemma enclosing the sarcoplasm. Each muscle fiber contains parallelly arranged myofilaments, which are known as myofibrils. Each myofibril has alternate dark, also known as anisotropic or A band, and light striations, that is isotropic or I band. This is due to the presence of 2 fibrous contractile proteins, that is thin actin filament and thick myosin filament. I bands contain actin, A bands contain actin and myosin, and they are arranged parallel to each other. A band bears a lighter middle region called H band. It is formed of only myosin. A thin dark line, M line, runs through the center of H zone. I band is bisected by a dense dark band called Z line. Region between 2 Z lines is called sarcomere. Sarcomeres are the structural and functional units of a muscle. I repeat, sarcomeres are the structural and functional units of a muscle. So let's see the structure of the contractile protein, that is actin. An actin filament is made of 2 filamentous actins which form double helix, as you can see in the figure. F-actin is a polymer of monomeric globular actins. Actin contains 2 other proteins, namely tropomyosin and troponin. 2 filaments of tropomyosin run along the grooves of the F-actin double helix. Troponin has 3 subunits. It is located at regular intervals on the tropomyosin. In the resting state, a subunit of troponin masks the binding sites for myosin on the actin filaments. Now, let's study the structure of another contractile protein, that is myosin. Each myosin filament is a polymer of many monomeric proteins called meromyosins. A meromyosin has 2 parts: heavy meromyosin or HMM or cross arm, that is globular head plus short arm. It projects outwards. Light meromyosin or LMM, that is the tail. The globular head is an active ATPase enzyme. It has sites for ATP. It has binding sites for ATP and active sites for actin.